Namaskar and good morning once again. Welcome to CEC Gurukul lectures. I am Dr. Kumar Shantanu from Department of Botany, Deshbandhu College. Till now, we have been discussing various aspects of Marchantia and its morphology, related aspects of anatomy and reproduction. We shall be continuing our discussion of Marchantia again. Till now, we have seen the morphology, various types of thallus organization, and now let us move on to the journey of anatomy of Marchantia once again. In case of Marchantia, the thallus has two distinct structures. One is called as entheridiophores, which is shown on the left side of the screen, and archegoniophores, which is shown on the right side of the screen. The thallus of Marchantia bear entheridia, the entheridiophore bear entheridia, and the gametophore bear archegonia. The two types of sex organs are born on different gametophytes, and hence Marchantia has a separate male and female plants. In Marchantia, entheridia are also produced in stalked receptacles known as entheridiophores. The type genus Marchantia is represented by about 65 species, of which Marchantia polymorpha is the most widely distributed. As far as the morphology of Marchantia is concerned, in mature thalli of Marchantia, the entheridiophores and the archegoniophores, which bear entheridia and archegonia respectively, are present at the growing apices of certain branches. Here you can see in the diagram that a male thallus will bear a male receptacle which is standing on a stalk, which is called as entheridiophore. The disc-shaped structure of male receptacle will bear numerous entheridia on its surface. The position of entheridiophore is always at the apical position, apical portion of the growing branches. Similarly, in a female plant, the archegonia is born on a female receptacle which has a stalk-like structure connected all the way to the thallus. These female receptacles, they bear archegonia on its surface. As far as the anatomy of Marchantia is concerned, let us discuss how it looks like from within. Internal structure of the thallus of Marchantia show high degree of internal differentiation and is divided into three distinct zones: the epidermal zone, upper or dorsal assimilatory zone or photosynthetic region, and the third, lower or ventral storage zone. In a diagrammatic representation of TS of thallus of Marchantia, you can very well see the uppermost region, which is the epidermal region. Below the epidermal region lies the photosynthetic zone, which is made up of parenchymatous chlorophyll containing cells. These are arranged longitudinally and looks like filaments. So these are filamentous in appearance. And at the ventral side we have lower epidermis and a storage region. A storage region has parenchymatous cells compactly arranged, storing all the photoassimilates coming from the photosynthetic region. The epidermal region consists of a single layer of upper and lower epidermis. The cells of epidermis have thin inner walls and thick outer walls and possess a few chloroplasts. The outer wall of the epidermis is resistant to water, checks the rate of transpiration, and thus is protective in function. The upper epidermis also shows presence of air pores or areoles. Each pore is surrounded by 4 to 8 tiers of 4 to 5 cells each, and these tiers are arranged in rings one over the other and form a chimney-like structure around the pore. The upper and lower rings of the pore are narrower than the middle, and opening is slightly raised above the upper epidermis. The cells of lower ring sometimes are papillos, project into passage in such a manner so as to give a star-shaped appearance. Each pore opens into an underlying air chamber. Unlike stomata, these air pores do not open and close, but carry out gaseous exchange during photosynthesis and respiration, and are thus analogous to the stomata of higher plants. In the diagram, you can see a schematic representation of air pore, which is formed by concentric ring of multiple cells, and in a transverse section you can see the pore in sectional view, and in the other diagram you can see air pore from the upper and the lower view. The photosynthetic zone in case of Marchantia lies below the upper epidermis. It consists of a row of small distinct air chamber. Each air chamber is bounded by one cell layer thick chlorophyllis partitions and communicates with the exterior through a barrel-shaped pore. Several short, simple or branched filamentous of oval cells rich in chloroplast arise from the floor of the air chamber. Each areole in the upper epidermis corresponds with an air chamber below. Since the cells of air chambers are chlorophyllis, this zone is the principal seat of photosynthesis in Marchantia. Here in this diagram, you can very well see the transverse section of a thallus, which shows three distinct regions: the upper epidermis and the lower epidermis, which forms the epidermal portion of any thallus; the photosynthetic region, which has filamentous cells and branched ones also. So, the chlorophyll containing cells are filamentous. They are arranged in a linear sequence, sometimes branched. The upper epidermis is interrupted by air pore, and in one photosynthetic chamber one air pore is present. So, each air pore represents one photosynthetic chamber lying below it, and below this photosynthetic region of Marchantia thallus lies compactly arranged parenchymatous cells, also called as storage zone. They sometimes carry oil bodies also, and mostly they contain the photosynthates which are formed by the photosynthesis by these chlorophyll containing filamentous cells. From the lower epidermis of Marchantia thallus arise two different types of rhizoids. One is smooth walled and the other one is tuberculate rhizoid. The two different types of rhizoids perform almost similar function, but tuberculate rhizoid is supposedly absorb more amount of water, as the invagination of the plasma membrane in tuberculate rhizoid makes the surface area longer. Also, from the lower epidermis arises scales which are multicellular in nature. The storage region lies just below the photosynthetic zone. It is made up of thin-walled parenchymatous cells and makes up the major part of the thallus. The cells are polygonal, mostly colorless, and are compactly arranged. These cells have the major function of storage and have starch, mucilage, and oil bodies stored in them. The midrib region is the thickest part of the storage region, but becomes progressively thinner, 3 to 5 layers, towards the edge. The cells towards the ventral surface have thicker wall and is bound by the lower epidermis. The lower epidermis shows two kinds of appendages, that is scales and rhizoids. The rhizoids assist in absorption of water and minerals, whereas scales help in retaining the moisture that helps Marchantia grow under dry conditions. The thallus of Marchantia grows by division of a group of meristematic cells which are present in the horizontal row just behind the apical notch. The cell division in this group of meristematic cells result in the increase in the length of the thallus lobe. Frequently, some of the median cells of the horizontal lobe of apical initials divide vertically to form a tissue that separates the growing point into two sets of apical initials. Subsequently, each set functions as the growing point of a new thallus lobe. This is the initiation of dichotomy and becomes marked by the addition of more and more tissues between the two sets of apical initials. Here in this diagram, you can very clearly see the apical notch where the meristematic cells are found. These star marks are the ones which represent those areas where we could find these meristematic cells. Now let us talk about the reproduction in Marchantia. Marchantia reproduces vegetatively as well as sexually. The vegetative reproduction in Marchantia can occur in 3 different ways. The first and most prominent is fragmentation. In aging thallus, the thallus starts generally disintegrating from the base, and when this reaches the point of dichotomous branching, the two lobes separate and with the help of apical growth form 2 different thalli. Here in this diagram, you can very well see that the older thallus starts disintegrating. The basal area gradually decays, and when this decay is complete, the upper branches, they get separated, now called as fragments, and thus each of these branches or fragments will be forming separate thalli. The second vegetative mode of reproduction in Marchantia is with the help of adventitious branches. Generally, these adventitious branches arise from the ventral surface of the thallus, though it has been reported by various authors that nearly all parts of the thallus are capable of giving rise to adventitious branches, but in case of Marchantia palmata, stalk and disc of female gametophore is known to form adventitious branches. So these adventitious branches detach and develop into a new individual as the tissue connecting them to the thallus decays. The third and most common and prominent method of vegetative reproduction in Marchantia is by formation of gemma. Gemma are stalked, disc-like multicellular structures produced inside specialized cells called gemma cups or cupules. Gemma cups are shallow, cup-like structures with fringed margins. Here in this diagram, you can very well see multiple such cupules or gemma cup arising on the dorsal surface in the midrib region, and each of these cups or cupules bear multiple numerous gemma. The gemma develop on the dorsal surface of the thallus around the midrib region. As discussed earlier, gemma are biconvex, disc-like multicellular structures with two prominent notches opposite to each other. In each notch is present a growing point. From these growing point arise two separate thalli. The gemma are borne on a short single stalk attached to the floor of the gemma cup. Several gemmae can be seen in surface as well as sectional view attached to the base of the gemma cup with the help of a single celled stalk. In addition to gemmae, the cup also bears several short granular hair that secrete copious mucilage. This secreted mucilage helps mature gemmae to absorb water and get detached from the base of the gemma cup. Gemma are carried away from the parent thalli by the action of water droplets, and they give rise to new thalli under suitable conditions. The mucilage which has previously surrounded each of these gemmae, they help in the retention of water. So even if the availability of water is little less, they suffice for the requirement of water for germination of these gemmae. In a transverse section, a gemma cup will look something like this. A transverse section or the vertical section of a thallus will show a gemma cup in which gemma is also surrounded by small mucilage producing cells. The gemmae arise from the epidermal cells on the floor of gemma cup. An epidermal cell becomes papillate and functions as gemma initial. At maturity, each gemma is a multicellular, biconvex, bilaterally symmetrical disc-shaped structure which is vertically inserted in the gemma cup with one-celled hyaline stalk. Each gemma has two growing points, one in each of the two lateral shallow notches. Most of the cells of gemma contain chloroplasts, but the marginal cells contain oil bodies instead of chloroplasts. Here you can see in this diagram multiple oil bodies on the margins of gemma. These oil bodies help in nutrition of growing thallus. They are the stored food material for the growing thallus, which will come out from two opposite notches of each of these gemma. Many colorless and densely protoplasmic cells are present on both flattened faces, and they are slightly larger than neighboring cells. These cells are known as rhizoidal cells as they form rhizoids on germination. Some club-shaped hairs present on the floor of the gemma cup secrete mucilage. This mucilage swells on absorbing water and causes the gemmae to break away easily from their stalks. The detached gemmae are finally washed away by the raindrops. The gemmae are also detached by the pressure exerted by the growth of new gemmae. When a gemma falls on the soil and conditions are favorable for its germination, the rhizoidal cells in contact with soil form rhizoids. The apical cells in the two marginal notches become active simultaneously and form two young thalli growing in opposite directions. After some time, the central part of gemma disintegrates, resulting in the separation of two new thalli. They produce more rhizoids from the lower surface and grow into adult thalli. Now let us talk about sexual reproduction in Marchantia. In Marchantia, Antheridia and Archegonia are born on special erect branches of the thallus called antheridiophores and archegoniophores respectively. Marchantia is oogamous, and sexual reproduction in Marchantia is dioecious, that is, male and female plants are born on separate thalli. These erect sexual branches are continuous of the thallus and grow vertically upwards through the notches at the end of the prostrate branch. An antheridiophore consists of a stalk with an 8-lobed disc at its apex. In fact, it represents a much modified branch system in which each lobe is comparable to the apex of a branch. This disc is formed as a result of repeated localized forkings of the young antheridial branch. Here in this picture you can see the antheridial branch, the antheridiophore which has a disc-shaped structure. This disc-shaped structure has 8 lobes. On these radial 8 lobes, antheridia are born. In a transverse section of antheridiophore, we can see the dorsiventral symmetry, typical of the thallus. The side corresponding to the ventral surface of the thallus usually has 2 deep furrows containing rhizoids and scales. This also tells us that the antheridiophore is merely an extension of thallus, which was earlier a prostrate one. In case of female sex organ, the middle of the terminal disc in case of antheridiophore has anatomy similar to the thallus, with an upper epidermis interrupted by barrel-shaped pores that open into air chambers containing branched chlorophyllous filaments. In addition to air chambers, there are many flask-shaped cavities which also have openings on the upper surface. Antheridia are produced inside these cavities. Each growing point of the disc produces a number of antheridia in acropetalous manner. A mature antheridium consists of a short stalk and a globular body. The jacket of the body is formed by a single layer of thin-walled cells and it encloses a large number of androcytes. When water enters into the slightly concave disc of antheridiophore, it moves through the narrow canal into the antheridial cavity. Now some cells of the upper portion of jacket of antheridium disintegrate. The androcytes come out from the dehisced antheridium and formed biflagellated antherozoites. A mature antheridium consists of a short stalk and a globular body. The jacket of the body is formed by a single layer of thin-walled cells, and it encloses a large number of enterocytes. When water enters into slightly concave disc of enthydiophore, it moves through the narrow canal into the enthydal cavity. Now some cells of the upper portion of the jacket of the enthydium disintegrate. The enterocytes come out and formed from the dehisced enthydium. So till now what we have seen is the structure of enthydiophore, the position of enthydria on the enthydiophore disc. In our next lecture, we will continue our discussion on the reproduction of Marchantia, and we will see the position of archegoniophores and the position of archegonia on the archegoniophore. Thank you so much.